All right. Hello, everyone. Welcome to the 5th episode of Future of Foraging. This episode is Follow Your Nose, Olfactory-driven foraging in mice and flies. We have two speakers, Venkatesh Murthy and Thierry Emonet, and one guest discussion panelist, Chris Rogers. All right. So do you want to start, Rory, with introducing Venkatesh? Venki?
Sure, I will. Yeah. So hi everyone. I'm very happy to introduce Venkatesh Murthy, our first speaker for the day. So Venkatesh began his journey in science with a BTEC in mechanical engineering from the Indian Institute of Technology in Madras before moving to the United States, and an MSE in bioengineering from the University of Washington, Seattle, ignited his interest in neuroscience and led him to his PhD in biophysics and physiology, also at Washington, during which he discovered his passion for cellular level analysis of neurons. And following a postdoc at the Salk Institute for Biological Sciences, in 1999, he began an assistant professorship at Harvard University, where he now works as a professor of molecular and cellular biology. And Venkatesh's lab studies the neural and algorithmic basis of odor-guided behaviors in animals. And they harness electrophysiology, optogenetic manipulations, and optic imaging to understand how the brain of mice reacts in approximated natural settings. And this allows their relation of neural activity to aspects of olfactory-guided behaviors, allowing inference into their computational and algorithmic underpinnings. So, very happy to welcome Venkatesh onto the stage, and the mic is yours.
Oh, okay. I was thinking I will introduce Terry first.
We can also do that. Sorry, you go for
it. Yeah, we usually do both in the beginning.
Yes, I forgot.
All right. So Thierry studied physics at the ETH in Zurich in Switzerland before pursuing a PhD in theoretical astrophysics at the University of La Laguna in Spain. He then moved to the US for a postdoc at the National Center for Atmospheric Research in Boulder, Colorado. And it was only after that, in 2002, that he switched to working on biological questions, as he discovered an interest in understanding how biological systems perform computations in dynamic and fluctuating environments. His lab at Yale now studies biological computations that aid organisms to sense and navigate their chemical environments, with the goal of understanding underlying computations at the behavioral, molecular, and cellular level. Thierry's lab works with a variety of models, such as bacteria, eukaryotes, and insects. And today he's going to talk about olfactory navigation in fruit flies, I think. All right. With this, we'll start the talks.
Great. So I'll share the screen, and I'm going to assume things are working unless I hear otherwise.
That sounds good. And I will switch everyone's video off.
Yeah, I think you need to do that for me to share.
I think you can just start sharing and it will automatically.
So is this okay now?
Yeah.
Great. Okay. Well, thanks everyone for watching. I can't unfortunately see you. I wish I could, but I'll assume that everybody's smiling and enjoying it. So I'll tell you a little bit about odor-guided navigation in terrestrial animals, essentially all behavioral work. And in fact, I would even not qualify this as work, because I feel like we will start off by just sort of defining things and talking a little bit about the bigger picture. I want to just acknowledge people over the years who have contributed, people in the lab, undergrads, postdocs, grad students, and wonderful collaborators. So odor-guided behavior in animals, I have 5 examples here. So I recently learned that certain kind of vulture.
Sorry, excuse me. We don't see your slides right now. In case there should be slides.
There should be slides, yes.
Okay, no. Right now it's just you.
This is always, gosh, let's see.
It's coming up. So
it came and it kind of did its thing. So anyway, so now?
Yeah, perfect. Now
okay, so this is an important slide that I want to make sure that this is all the people in the lab who done all the work, and collaborators and funding sources. So this is a slide with a few animals, where the first one is this kind of vulture called turkey vulture, which is unusual in having a large olfactory bulb and olfactory system. And this is thought to be related to the fact that it forages in very dense, they're looking for cadavers and carrion in dense areas where visual information is probably not as good because it's all hidden. So that's very large scale, meters and maybe miles. This is, of course, a famous moth, different kinds of moths, and this one that's taking nectar from the datura flower. And their the olfactory guided navigation has been actually really wonderfully studied at the ecological and behavioral levels, as well as the neural level. This is, we're again talking in the meters and maybe a little bit more. Just an example, crustacean in water, so a different kind of fluid dynamics, but just maybe it's a matter of scale. They too have been studied for odor-guided navigation. And then the other thing that we've gotten interested recently, another thing that one studies, is navigation and terrestrial on the ground. And this is a little bit different because we have stuff stuck on the ground, but it's maybe patchy, and then animals follow this. So this is actually from a review that should appear in the coming year, largely written by the physicists, Gautam and his advisor Massimo Vergasola. And I was part of this, where we sort of point out that, and I think Thierry will also discuss this, that basically odor landscape, the physics of this really should be governing the search strategies. So in the case of bacteria, we're talking about microns, tens of microns, where you have chemotaxis, and as a function of time, if it's a successful gradient climb, you would imagine the concentration to fluctuate, but it's relatively smooth all the way through. Similarly, at short distances in airborne situations where, let's say, there's an odor source, I'm pointing to my mouse. I hope you can see it moving. So in this case, in the shorter distances, there are fluctuating, so-called plumes of odors advected typically by wind, because diffusion is too slow. Again, at these distances, under many conditions, the concentration of the odor the animal will encounter, yes, it's fluctuating, but you more or less get continuous encounters. This contrasts with situations, which I think Thierry will also talk about, where at larger distances or at short distances with very large Reynolds and Peclet numbers, you will have large pockets of space where there is no odor. So you have these infrequent, intermittent encounters. So the strategy in this case, and I think for completeness sake, let me also talk about trail tracking. You might imagine this is a much more smooth, trail track. How hard can it be? But you can see from the way animals track trails, again, the encounters with the odor, the concentration is also going to be highly intermittent. Unclear it will be as intermittent as these flying insects, for example, but this is also, you go off the trail, nothing on. So in general, I think these different kinds of landscapes, I think if we were to make one very qualitative observation, which can be, of course, quantified, it really seems to boil down to the fact whether the strategy should be just reactive. You encounter something and off you go. You're reactive. Or you really need to take history into account, planning. And this is perhaps very easily understood here in the case of flies. In periods when you don't have a lot of encounters, if you didn't know anything about what was happening before, you're in much worse off shape than if you're just waiting for something to happen, right? And this can be quantified. This has been quantified, at least in the theoretical conditions. And moving forward, I feel strongly that thinking about this in experimental design is extremely helpful, will be extremely helpful. So that's the big picture. So I want to actually illustrate 3 different situations, and I will maybe focus on the first, second, and leaving the third largely to, I think, Thierry has done some beautiful experiments on this. And this is airborne cues at short distances. We're talking about trail tracking, and at larger distances, airborne cues. And I will illustrate this with experiments from our lab, but I want to acknowledge that there have been many studies by others that are done. For example, for airborne cues at short distances, this is by no means exhaustive, but a few recent ones include one from Upi Bala's lab. This is our lab. This is Kathy Nagel. Nathan Urban and Matt Smear, and then there's exception to this short distance but yet very fluctuating is Thierry's paper. Okay, what did we do? This is a few years ago. So in this case, we wanted to basically ask and demonstrate whether mice can use airborne cues to find the source of the of this airborne odor cue. And at that point, in fact, it wasn't clear, and we were in fact chided in grants that the claim was that everybody knew that mice don't use airborne cues. They just scurry on the ground. They use terrestrial cues. So we were curious. The experiment was relatively straightforward. We had basically a flow created by a small computer fan that just kind of flowed through about a meter square box arena. This is very similar to all the hippocampal studies, for example, for place cells. The mouse is just running around in the dark. It's not quite dark. It's far red light, infrared light. 3 odor ports, this one, or this one. Let's ignore this for now. So the odor is released from either of these ports, and if the mouse directly approaches that, not checking the other ones first, then the animal will get water, and they are water restricted, so they're thirsty. So they will do this. And the animals do this extremely, very quickly. Within a couple of days, they learn this. They clearly use the airborne cues. We've done many controls. And I think our conclusion was in this little graphic from that paper, where we found that with limited experience of the knowledge of the source locations, this is early in the animal's training, animal clearly seems to navigate, if you will, follow the odor cues, the airborne odor cues. The other interesting aspect of the study was that upon repeated training, meaning exposure in many days, animals really know the location of these things, and they basically just forage. They just go one by one, and that we could formally show that that was a better strategy. And this also happened because we relaxed this constraint that they can arrive at the target no matter where, and then they will get the water. This part I'm not going to tell you about. For this gradient, for this finding the source with airborne cues, we showed essentially with calculations, with the model and with calculations, and showing that the behavior of the mice were predicted by these calculations. And the idea was rather simple, that if you were a searcher, and if you measure the local gradient in concentration, let's say there's a method where we didn't even say how the mouse is supposed to be calculating this gradient. The point is, if you are in a particular place, and if you calculate the local gradient in the concentration of that odor, and let's say that is pointing in this direction in this case. This is the size of the gradient and the direction the gradient is pointing to, but the actual source, let's say, is in this direction. And the reason the gradient was like this because of the fluctuations, right? If there was no fluctuations, if it's completely slowly diffusive, the gradient will always be pointing to the source. But because of the fluctuations, the gradient points. In another situation or another spot, another time, perhaps the gradient is here and the source is here. So if upon repeated measurements, if you have more than half, ideally much more than half of your estimates of gradient is pointing towards the source, meaning that you can think of it as the dot product or the projection of this vector on that. Either way, if there's enough of this delta phi that are less than pi over 2, and the more times they are, the quicker you'll climb up the gradient to the source. So this is just simple understanding that as long as you have enough of these gradients in different points, that you should, over time, to allow you to point to the source, you will eventually get to the source. And I think that's, in this situation, it's almost certain that mice are using some sort of average gradient ascent to get to the source. Of course, this works if the gradients are actually informative. If it's highly turbulent, most of the time, or at least with equal probability, the gradient is going to point away from the source as it is towards the source. And therefore, the mouse is, it's going to be more or less chance to get to the source if you just use gradients. So that's one, I guess, little insight. And I think there are many experiments by several people doing these sorts of, looking at this particular aspect of short distance, smoother gradients, and how mice or other animals go up the gradient. Another situation is this trail tracking. There are far fewer studies in order trail tracking, at least in recent years. There's Of course, historically, there's many behavioral studies on dogs and other cases. There's, of course, this famous study by Noam Sobel, where there were Berkeley undergrads, humans tracking chocolate trails on grass and so on. But recently, Ubi Bala's group, and I think we did one on mice, and then there's one in Nathan Urban's lab, and we have a very exciting study in progress. Hopefully, we'll see this out soon. So tracking trails, scent trails, just random search will tell you that many animals do this. So this is our study in carpenter ants published a few years ago. So what we did was to isolate pheromones from the gasters, just draw them on paper, and this is again infrared light, so the ants are not able to see this the color, but they're obviously smelling the pheromone, and we just watch them follow this trail. And 2 antennae are marked, actually in this case manually marked by an undergrad, frame by frame in MATLAB or so. And the point is that they can do this behavior very well, and we can study this in a very controlled, and we hope it's relatively naturalistic in terms of freely moving and so on. And we learned a lot about how ants use antennae to do this trail tracking. And I just love this close-up view. This was actually in the published paper. Here in the close-up view, you can see, let me stop this so it's less distracting. And it's slowed down quite a bit. And I'm always amazed by how much, there's just a lot of things going on than just simple lateral gradient or whatnot. There does seem to be some version of planning and history dependence and so on. Okay, so we did that, and that was published, so I'm not going to tell you much more about it. So we've been working on it's a similar thing inspired by the work of Khan and Bala, where we have an infinite paper treadmill on which mice run. This speed is controlled by a DC motor. Originally it was open loop, but now we can actually do closed loop control of the speed of the paper. We hijacked a print head from an inkjet printer where we could then print odor. So here you can basically load up some odor that you care about, and print it on this paper as the paper is moving. Hopefully in this GIF you can see that. Somehow that didn't show here. So you can see that this printhead is printing the odor trail here. The paper is being fed into this arena in which the mice are going to be tracking those odor, or we want them to track the odor trails. The beauty of this is you can use this inkjet printer. You can send a control command to this to draw almost any form of the trail. In principle, you can put multiple printer heads to print multiple trails. And the the free form and continuous form of this, I think, makes it very unique to study this as a sort of a virtual navigation, if you will, odor guided navigation. So here's the video. This is already old by now. It's 3 different views, 3 different cameras, and the trail is barely visible. There are bits here that may be slightly visible. The paper is moving in this direction. The trail is sort of coming through there, and the same thing here. And there was a drop of actually chocolate milk where that's the reward for the mouse following the trail, and they would stop and get dragged behind. With closed loop, we could in principle stop the treadmill during the drinking of that, and when they're done, we could start the treadmill up and so on. Okay, so we could study this trail tracking behavior, and I think I don't have to tell the neuroscientists among you that in these kinds of arena, of course, it's decades-old technology now where you can put things like tetrodes, or these days, of course, other higher density electrodes, or even mini microscopes and so on. So the neuroscience part of it, I would argue that technically it's really well within reach, and it's really up to us to come up with very clever behavioral paradigms. So just one as an aside, this trail tracking thing was actually a big motivator for Alexander Mathis, who was a postdoc in my lab at the time, to start with Mackenzie Mathis, this whole deep lab cut thing. So what's the big deal about tracking scent trails? I mean, I see the trail and you just follow it, right? And I think for those of you, I'm sure many of you have thought about this. For those of you who perhaps haven't, here's, I think, a helpful intuition. If you're using a visual trail as an analogy, imagine you're hiking, walking along this trail, you see it. Yes, of course you see it and you could see the whole thing. If you're here, you see the entire trail and you can plan it. But imagine it's a super foggy day right? And you go there and the trail stops. So you have the history of what you've done. Now you have to make a best guess as what's next. That's exactly what scent trail tracking, because scents, you're only smelling what's right in front of you, or the animals only smelling what's right in front of them. So you have all the information in the past, if you could, store in the brain, and up to now, and then you have to make a projection as what's next. That's what makes scent trail tracking a very interesting problem, non-trivial. So our collaborative, I didn't contribute this other than just to discuss with them. This is again Gautam Reddy, Boris Schreiman, and Massimo Vergasola. They have a recent preprint with, I think, is absolutely beautiful normative theory of how to think about trail tracking. If you were an agent, let's say that this is the true direction of the trail, and your sensor is of certain size A, and it's, let's say, the last detection was here. This is where you last detected the trail. And then here you are now, and you have an estimated trail heading from that. And so with all of these parameters, they have essentially a theory to say, what should an agent do given the past sensing positions? Because the sampling is discrete, right? I mean, you're not getting samples all the time. It's always discrete, either because people like us, animals like us, we sniff, or for ants or others, they're moving their antenna and you're encountering or not encountering. So it's discrete. Based on all of this, the animal will have to now make an estimate of the heading plus the associated uncertainty. So it's essentially a sector search strategy, as they call it. And in the paper, they're using the strategy and a reinforcement learning agent with the entire Bellman equation and everything. They can show that they can reproduce experimental trails from, this is Khan and Bala with a broken trail for a rat, and this is our data on an ant sort of going around kind of a curved pheromone trail, and their agents basically will do that. So they will track, and then once you lose it, you would have this increasing casting size until you encounter. With two sensors, you would see this kind of alternation of going back and forth. Formally, of course, every theory you could argue is going to be wrong, but I think these kinds of formulating the problem in a very normative way allows us to say, what are the key things? What are the experiments one should do? And I think we are very excited to move forward to do experiments with our treadmill in mice as well as in ants in an arena. Finally, I think let me just take maybe a few minutes to tell you a little bit about this last problem, which I think Thierry will tell you in flies. And the idea is that at long distances, things are extremely fluctuating and intermittent. There are, of course, many beautiful classical studies in insects, including David et al. In Nature, Ring Carter's group, and Jeff Raffel. And also recently, there are forays into mice, not about the actual navigation, but how mice can take care of these very fluctuating intermittent cues. And I'll tell you a story that we are actually converging on in our group. And this picture, I suspect Thierry will also show to indicate that, like, as a function of distance, right? I mean, the actual the advected or the transported odor is going to be highly fluctuating. And just to make a point that it's not only large distances formally, but you can also get at short distances under certain conditions. And this is the moth, the tracking, where you have very characteristic sort of casting behavior when there is no encounters. When there's encounters, you kind of go upwind, or the moth goes upwind. And I will also mention one strategy, and this is a purely sort of a theoretical strategy, and yet to it's unlikely, I would say, in the formal sense, it's exactly implemented in animals, but infotaxis or search, going up information gradients under these conditions. So the 5-minute bit of story that I want to tell you from our lab is based on this observation. The farther you are from the source of odor, in these kind of highly turbulent conditions, the odor encounters, so this is a function of distance, the encounters, the frequency of the encounters of these odor packets, if you will, gets less and less, or they get more and more intermittent with more inter-packet intervals. The closer you are, you still are getting very fluctuating encounters, but they're much more frequent. So if you have the luxury to wait and integrate over time, there is some information about the distance of your location to the source based on the relative sort of intermittencies across the two. So we asked a question, and this is almost purely like what if question, whether mice, completely head restrained and highly artificial in this sense, whether they can tell us whether over a period of time the encounters of these odor packets are coming at a high rate or at a low rate. So we trained mice to sample brief pulses, and we can create pulses as thin as about 40 milliseconds. And we repeat them in a pseudo-Poisson rate, let's say a Poisson rate, a pseudo-Poisson process format over a period of a few seconds. And if it's a high rate above a certain threshold, the mouse has to guess, it goes left. And if it's high rate, it goes right. Whichever way, like one at the high and low. And it's basically a two-choice experiment, high and low, and each of this is coming from a distribution, and from trial to trial, it's fluctuating. And you also get mistakes, and they get punished for that, and with a longer timeout. Again, to make clear the logic, that if the odor source is further from the source, we're mimicking that with higher Poisson rates of these odor pulses. Closer to the source, they are lower. The other way. Closer to the source would be higher rate. Further from the source is obviously lower rate, because the further you go, it's more intermittent. But the bottom line is that mice can actually learn this rather quickly. So as a number of sessions, this is performance. Two choice, so 50% is chance, 0.5 is chance, 1 is perfect performance. With the brief pulses with incredibly high contrast, that's a straightforward two choice, like basically order there or not. And that animals can learn in a couple of sessions. We keep increasing the duration of the integration, and in parallel also decrease the contrast. And we can show up to 3-to-1 contrast, meaning let's say 4 pulses in 5 seconds versus 12 pulses in 5 seconds, coming at random rates, again, at random timing. The rates are the same, but they're coming at random timings. And mice can actually do very well, like 85 to 90% performance. So mice can learn this. And I think, we also saw parametrically that the mice are actually integrating these pulses over time, because as you increase the sampling time with lower and lower contrast, they do better. Because if you have very low contrast, meaning only 2-to-1 ratio of the pulses, there's going to be a lot of mistakes when you're only integrating for a brief period of time. But given long period of time, you can integrate enough to know, yeah, that's high, this is low. So this experiment essentially just tells us that mice are, in principle, capable of sitting in one place and sort of integrating the information of these pulse rates and making guesses to how far they are, if they are farther or closer to the source. I have no idea whether mice in real life use this kind of a strategy, but at least we wanted to know whether this information is even possible for mice to take advantage of. So let me actually just close there by just putting this summary slide up and then saying, I think one of the driving, themes of, at least for our lab, and I think many people we talk to, is that the strategies that animals use to use odor cues to navigate, if that's what we want to call it, is going to be highly dependent on the landscape, whether it's short distances, whether it's diffusion related versus transport that's more smooth, fluctuating versus transport that's highly, highly intermittent versus its trail tracking. So let me stop there and hand this over to Thierry. Thank you for listening.
We have time for just a couple of short questions, that if there are specific questions to your talk. We got a few. So I'll start with one from Ashley. This was, I think, about the trail tracking experiment. And she asked, have you controlled for texture in this instance?
We have not explicitly controlled for texture, but we know that if we put just the ink, so the color is basically a fountain pen ink without the pheromone, ants won't track it. So there's no extra texture created by the color, and the pheromone is extremely dilute, so I can't imagine that adds some texture. It's a great question. Yeah.
And then another question from Matt. This was about, I think, that mouse experiment with the paper roll. Wait, oh God, sorry, now it moved. Sorry if I missed. I see. Oh God, no. Sorry, the questions when they get voted. Oh God, I don't know where else. Here's one from him anyway. How am I sniffing in your tasks?
Yeah. Sorry. How am I sniffing?
Sniffing in your tasks.
Which task? The head fixed fluctuate. So I can tell you for both. So
I'm not sure.
The freely moving animals, we've had actually quite a lot of trouble getting consistent sniff recordings from that, because we wish not to put the standard thermocouple or this Matt smear. Hi, Matt. So we are not putting the like Matt himself has done, the thermocouples or the pressure thing, because we want to keep the cavity intact as much as possible for future. We're worried a little bit about interrupting, at least unilaterally. So we would like to avoid. So we've been trying various other tricks. We have a few recordings of the sniffs in this freely trail tracking one, but nothing to write home about. But in the head fix that pulses integrating, they were measuring using this mask that many people are using, like I think Kevin Franks, for example. So Matt's saying the thermistor doesn't penetrate. Yeah, so we can talk about this offline, but with the intermittent head fixed mice, we absolutely are measuring the sniffing. In the trail tracking, for most experiments, we're not, but we will really, really want to.
Okay, there's one. So that was the question I meant to ask initially, was how long is the paper loop, and does the trail repeat?
It does not, because it's an open loop. The paper is not circulating. It's to be very clear. In the drawing, I had it as recirculating. That's wrong. Sorry. It's basically an infinite paper treadmill. It never repeats.
From Marisol, we have a question, and they ask, would the mice be using the visual cue of the printed order trail?
So we think not, for two reasons. One, it's illuminated by pretty far red light. It's close to infrared. We don't quite infrared because our cameras are not that great in pure infrared. So we don't think that they're using stuff. The other way you can think is it's a little bit tricky to do the control experiment of not adding the odor, because the ink itself has a smell. So we have to rely on the fact that it's mostly in the dark, and that's the reason why we don't think. I have to think about other controls. Maybe I can answer you at the end of it if I can think of other reasons why we think it's not the visual cue.
Sounds good. So one question from Rory is, what is the ecological relevance of olfactory trails for mice in nature? I had assumed that most olfactory stimuli would tend to travel as airborne signal close to the source.
Yep. Yeah, I don't know. And I think this is one of the most embarrassing things for two reasons. One, because I am very poorly educated in ecology. And two, I just cannot find information, even with the internet, lots and lots of searches. I don't think there are very many studies on this. I don't think it can only be airborne, though, because they do deposit things. I mean, people imagine like they're doing urine marks and things like that, right? So I would bet quite a bit that they're going to use surface-bound cues. Whether they're in the form of trails is a great question. In fact, I don't know if other people can maybe chime in and write. I don't know what the natural or odor-guided behaviors in mice are. Like, who has gone and looked at? Just follow mice around as they are running around and getting in natural conditions. There's some old experiments on things like deer mice and buried seeds and squirrels and whatnot, but that's more statistical observations rather than watching them, use olfactory cues to get, just like we do for ants, for example.
Yeah, I think we could dive into this sort of ethology a bit more maybe at the end. Mm-hmm. This seems like a broader question. I'll just see. I'll ask. We have maybe more questions than we have time.
So that's okay. Yeah.
So from Dinesh, he says the intermittency test. How about humans? Do you hook it up and check how it feels? Does it does it feel like a concentration change, or does it require active integration?
Yeah,
if you hook it up.
It's a great question. So we're now actually struggling conceptually about how do we distinguish, like, is it really, as you say, is it some sort of an average concentration over time? or it really is it counting pulses, for example, like for example, the Brody task or something. Actually, in Brody, I don't think it's counting pulses either. It's probably also integration in some other way. But yeah, it's a good question. For humans, we're so bad at this. I don't think we should really necessarily count untrained humans anyway about this. So we're thinking about this, but I don't know the answer.
Okay, thank you. From Christina, great talk. Sorry if I missed this, but in the last experiment you showed, do you think mice count the peaks or integrate total concentration?
Yeah, I think we were just answering that, and I think
exactly.
we're converging. I mean, I can tell you without, and this is ongoing stuff, we think it's more towards counting pulses. I think in part because I'll give you one small reason. The olfactory sensory neuron responses are extremely transient. So there's no signal, as far as we can tell, leaving the nose that has the feature of an integration signal. That doesn't mean that the brain shouldn't interpret now perceptually, but we know the signals are leaving the epithelium in a very intermittent manner.
Okay, thank you. Maybe I'll do a last one and we can return in the discussion to, there are more questions. Yeah, I just don't want to run too much over time. So from Gonzalo, for the odor propagation in air, the odor plumes become sparser, but they also become briefer. How do you think does this put limits on the amount of time that the circuitry have available to decide that the encounter odor filament corresponds to the odor that is being tracked?
Yeah, it's a good question. So I don't remember off the top of my head, but these kinds of statistics were theoretically calculated very beautifully by Celani and Vergasola in the Physical Review X paper. And if I remember correctly, the most prominent feature that decays with distance is actually the interpulse interval, much less this whiff duration and then the kind of the height of the stuff. But I may be wrong, so I'm not remembering this exactly. But if it is true that not only that the interval gets longer, but maybe the individual ones are less bursty, it's a good question. So maybe I wonder if you could also use that as another cue to get a sense. So then I don't know the answer. I mean, there's very few experiments, certainly in mice, using very brief pulses, right? I mean, Andrea Schaefer's paper that just came out is one of the first, and we're actually also working on responses to these very brief, very filamentous cues.
Okay, thank you very much. We have more questions. I will. We can return to them in the discussion. Hopefully that sounds good. So I will. So I should have done this earlier. Need to invite Thierry back on screen. Okay. And then I will move you off screen and see you shortly.
Can you hear me?
Yes.
Yeah?
Yeah, great.
Awesome. And so now I share my screen. Select the window. It worked a few minutes ago, so
yes, this looks good. Okay.
Yes. Do you see everything? Good. Awesome.
Yeah, I think
so.
so.
Thank you very much for the invitation, and it's actually great to follow Venki because he already introduced a lot of the themes for me. The first thing I wanted to say is that everything I'm going to tell you about has been done by these two fantastic postdocs, Mahmoud Demir and Nirag Kadakia. And they're both looking for jobs. So if you're hiring, check them out. Here's the paper I'm going to talk about. And okay, let's see if I can pass my slides. Yes, it's working. So in my lab, we're interested in understanding how biological systems basically acquire information, process it, and make decisions. And as a framework to study this kind of question, we use chemical navigation because it's actually a very complex task. You have to detect sense the signal, adapt the sensor to the concentration so that you remain sensitive, process that information, make some decision, and then when you act on it, you're going to change your trajectory, and that's going to cause in turn to change the signal you're going to experience next. So not all of your behavior is about following the signal. It's also about gathering the signal or exploring to get the signal. And so we've done a lot of work with bacterial chemotaxis that you're seeing here. It's probably the best characterized signal transduction pathway in all of biology. We understand it from molecules all the way to behavior of populations of cells. And then we also study this similar type of question using fruit flies. And so this is an experiment I'm going to talk about that was mentioned by Venki, where those flies are hungry and they're following smoke. We found that they actually love smoke. And the nice thing about smoke is that you can detect it with the camera. And so this is an experiment done in the dark. The flies are illuminated in infrared, and you can see the signal. And so the question I'm going to ask today, or I'm going to discuss today, is what kind of strategy do the fruit flies use when they navigate such signal, where, as Venki was saying, the fly is really experiencing packets of odorant as it's navigating. So before that, I just want to give a little bit of a history about those questions that have been basically addressed by many before us. And I think Venki even showed the same plot. So there was this awesome experiment by Kennedy and Marsh in the 70s. What they did, so one of the big difficulty to figure out how animals or insects navigate odor plume is that you cannot really see the odor. And so they solved that problem by releasing a pheromone together with bubbles, soap bubbles. And so then this thing is dragged down by the wind, and they release a moth that navigates all the way to the source. And because they have the bubbles, they know when the moth is within this cone of the plume or when it's exiting this cone. And so the trajectory here is thicker when the moth is in the cone, and it's thinner when the moth has lost the plume, basically. And you see that when as soon as the moth loses the plume, it goes crosswind, trying to catch again the plume. And then when it's in it, it moves more upwind. And so there's this turning upwind when encountering the plume and casting crosswind when losing it. This strategy seems to be a very good strategy to navigate this type of plume, to basically stay inside the plume. Much more recently, Cathy Nagel studied fruit flies walking in a wind tunnel. And in this wind tunnel, she was able to basically generate blocks of odor. So at some point, she can turn on the odor, and the entire wind tunnel becomes odorized. And then when she turns it off, the whole arena becomes loses its odor. And so the fly is experiencing odor everywhere or no odor. So there's no real spatial structure in this odor, but she can control the temporal aspect of the odor. And what she found is similar to what I mentioned before, is that when the fly is in the odor, which is the pink trajectory, it's moving upwind. When the odor finishes, the fly starts to search, going across the wind and basically initiate a local search to try to find the odor again. And she was able to measure basically basic responses as a function of time, like the change in this upwind velocity, ground speed, angular velocity, and so on. And again, the basic strategy that the fly was using is to reflectively turn upwind when encountering the odor, but casting crosswind initiates a search when losing the odor. And then there are experiments using flies that are flying this time in a wind tunnel, so by Dickinson's group. And there they released a straight plume that they basically mapped out with a photoionization detector. So then you know that the plume is in the center there, and you can track the fly flying, and you know when the fly is entering or exiting the plume because the plume is always at the same position. And from there, also, they found that the fly was basically casting crosswind when it would lose the plume, and then going upwind when it encountered the plume. So, but there is another set of experiment by Mafraneto and Carde in the 90s, where they realized that the timing of the encounter with the plume really mattered, in the sense that if you do an experiment with a straight plume, with a moth, you're going to see a trajectory like that. But if instead you pulse the plume, even though it's straight, now it has moments where it's on and off and on and off. You see that the moth moves much more straight upwind. So it seems that the moth is sensitive to the timing of the odor encounters. And timing seems to be very important when you have to navigate a signal like this one, because you don't have much of a spatial information, because the gradients of the odor concentration can point in very random directions. So the information you really have is when is there an odor, is there no odor, no odor. So you get basically a time trace of the events of the odor. And so the question we wanted to ask was, how do odor encounters, these encounters with these discrete packets or ribbons of odor, shape the navigation of an insect when the plume is spatially complex and intermittent? So not only it's intermittent like that, it's changing on, off, and on, off, but it also has a spatial complexity to it. So that was the goal of our study. So now, in order to visualize the odor, we tried first to use fluorescent odorants and so on, but we had to go to such high concentration that they were toxic. And then we found that flies love smoke. And so you can image smoke. So this experiment basically has two glass plates separated by about a centimeter. And then there's a laminar flow coming down. And then in order to perturb that laminar flow, we have tiny little jets here that cause this straight plume to basically be broken up. So it's not a turbulent flow. It's a laminar flow, but it's very unsteady because the perturbation of the wind is unsteady. Okay? And the result is that you get these packets of odor that travel down. And so then the first question was, is the navigation really driven by olfaction? And so the first thing we did is to do those responses of the behavior of the fly. Now, using a plume that was straight, instead of perturbing it like we did in the previous experiment, we just let the plume being very straight. So in this case, the flies navigate by zigzagging through it, just like a trail navigation. And you can count the number of flies that is around the trail. And if you count the number of flies as a function of those, you see that it increases. And then you can do the same experiment. Instead of putting smoke, you put ethyl acetate or apple cider vinegar, and you get the same type of behavior. And then we run a whole bunch of control to show that this behavior is really olfactory. So if you knock out the CO2 receptor, they're still attracted by the odor. If you remove vision completely by having NORP-A mutants, then you see that the flies are still doing it, so they don't rely on vision to do that. You can saturate the entire system with humidity, they're still doing it. If you knock out ORCO, which is a core receptor for the olfactory receptor neuron that respond to many odorants, you see that in this case, the response is almost gone, but there's some sort of repellence. That's because CO2 is still being sensed, but now only CO2 is being sensed. If you knock out ORCO and these guys, then you get anosmic flies, and then there's no response. Okay? So now we have an olfactory response to a signal that we can see, so we can basically start to examine how the behavior depends on the signal. So to detect the signal to quantify the signal experienced by the fly, the difficulty is that you're seeing the fly and the odor in the same channel. So you cannot really measure exactly at the fly. So we integrate the signal in this pink area in the front of the fly. And then we have the speed, the orientation, and you see that the signal is very choppy. it has this burst of plumes going through. And so the fly is really experiencing these packets of odor that's flowing over it. No? And the first thing we did then is to examine what would be the behavior of the fly if we interrupt the odor and then start it again and interrupt. So we do blocks of 15 seconds, but we keep the wind, the fluctuating wind, the same at all time. So now you see for different trajectories of individual flies as a function of time, each little dark dot here is an encounter with an odor. And we can look at the probability to encounter odor. So you see it's on the order of between 10 and 15%. So it's really a sparse signal. It has low intermittency, if you want. But the fly responds very strongly to this. So the orientation probability to turn upwind strongly increase when the odor is on, and then goes back to basically crosswind when the odor is off. There is some modulation of the angular speed and the walking speed, but not very strong. And you see that another thing that is very strongly modulated is the probability that the fly will be walking or stopping. So the rate of transition from walking to stopping and stopping to walking is strongly modulated by the presence of odor. So there is the orientation and the walk to stop and stop to walk transition that seem to be responding very strongly. So we focused on those two measures of behavior. And the question we ask is, how do the orientation and the transition between stop and go depend on odor encounters? So for the orientation first, the first thing that you can plot is that you say conditional on the fly experiencing an odor at this time here, what is its orientation 2 seconds later? And if there was no other encounter with a packet of odor during these 2 seconds, this is what you see. So conditional on the orientation of the fly at the onset of the whiff. You see that if the fly was going upwind, it's kind of staying more or less upwind. Positive here is a positive bias will increase going upwind, a negative bias will increase going downwind. So 0 is kind of no change. So there is a response, but it's not very strong. But as soon as you experience more whiffs, the response becomes much, much stronger, and the orientation bias is strongly changing. If you go in crosswind, for example, you really reorient upwind. So this seems to indicate that the history of whiff matters, or the frequency at least matters, no? But it could be also that the duration matters, because you have let me put it. So one question we had is, do the fly care about the frequency, the duration, or the concentration of those other packets? So you could have the same different frequency but same duration, or different duration but same frequency in this case. And so which one is driving more of the response that we're seeing in those experiments? And so if you sort the data and bin it by whiff duration, so by durations, then you look at the dependency of the reorientation as a function of the whiff frequency, you see that there's definitely an increase in the orientation to go upwind as a function of the frequency. But if you do the same thing for the whiff duration, you don't really see a very strong dependency here. So one possibility is that our whiffs are really, really short, much, much, much shorter than, for example, the one that you can see in Cathy Nagel's experiments, no? And so maybe that's one of the reasons why in our experiment, it seems that the frequency is a better predictor of the change in orientation than the whiff duration. We also checked if the concentration of the odor, because as Venki was saying, as you get closer to the source, you expect a change in the concentration. So we regress the change in orientation with respect to the duration, the frequency, and the concentration. And you see that really the strong dependency is with frequency rather than the other two measures of the odor. So the frequency of odor encounters really drives upwind turning in this experiment. Now, what navigational behavior drives this upwind bias? Like, how do they actually bias their motion upwind? So one thing that's interesting is that if you look at the orientation of flies, you see that it has this abrupt change, like saccades. And there was a paper before us that showed that fruit flies tend to, when they're walking, do saccades of plus minus 30 degrees. And so we looked in our data and we found exactly the same type of saccades. And so we were interested in what is the rate with which the flies instantiate those saccades, or what is the time interval between saccades. And you find that what we observe is that this discrete turn obeys some Poisson process. And surprisingly, this Poisson process doesn't seem to be depending on the frequency or the duration of the encounters with the odor, or low frequency, high frequency, short duration, long duration. It seems that you always get this same distribution of time to turn, basically the interval of time between two turns. So it looks like the flies are basically instantiating these random these turns at random time, but with the same exponentially distributed distribution. But if you look at the direction of the turn, those are biased. So even though the fly instantiate turns, it tends to bias the turn upwind more as a function of the frequency. So when the frequency of whiffs gets high, the angle tends to be very much more biased upwind than downwind. So you can recapitulate this with a stochastic model of turning, where basically the fly is doing a bit like bacterial chemotaxis. The bacteria instantiate these tumbles at random time. And here, the fly is instantiating those turn at random time, but its probability to turn upwind, given that it's turning, depends on the frequency of the whiff that it has experienced in the past. Next, how do the transition between stop and go depend on these odor encounters? So to examine that, what we did again is these blocks of 15 seconds with odors, 15 seconds without odor. And you see here that when there's odor, the flies are moving very much. And then when the odor goes away, a lot of the flies stop. And so we were interested in what's the, how long does it take for the fly to decide to stop? How long does it take for the fly to decide to stop to go again? And so we're looking at, and the walking speed, again, doesn't really change very much. But the transition between walk and stop do change a lot between those. Okay. So if we look at now how the decision to stop, like how long does it take after I experience a whiff for the fly after the fly experience a whiff, how long does it take for it to stop? So if you look at any whiff, you get this distribution here. If you randomize the times, you get this other distribution. But what's interesting is to look at what happened when there is a whiff and then no more whiff. How long does it take for the fly to stop without whiff, following the last whiff, if you want? And this is this purple distribution. And then we wanted to know, well, does the history of whiff before the last whiff matter? So maybe if the fly was experiencing a lot of whiffs and now it hasn't experienced a whiff for a little while, its decision to stop might be changed because there was a lot of whiff before. And it's not. So it seems that the decision to stop basically only depends on the time of the last whiff. And you can recapitulate this with a model. So you can say, we built 3 models, and then we compared all 3 to the data, and the only one that really worked was this one. So this model has this probability to switch from walking to stopping that drops at every whiff onset to the same value and then relaxes. okay? But like this model accumulates evidence. So every time there's a new wave, the thing drops more and more, which means you accumulate evidence. And this one is just basically based on the duration of the waves. And what we notice is that really only this model could replicate the data. And so this is the prediction from the model. So this is what the model predicts, and this is the data. And you have this, as the wave frequency increases, the duration of the walk basically increases. And that's also what you see in the data. Now, so the bottom line here is that the decision to stop is just based on the time to the last wave. Now, what about, oh, and yeah, I just showed that. So then the decision to go, so now the fly is stopped, hasn't experienced a wave for a little while, and how does it decide to go ahead? And here you see that it's different. There's a difference between the purple and the green curve. So there's a difference between whether the fly is experiencing a clump of whiffs or multiple whiffs or just an isolated whiff. And again, if you do multiple models, this time the only model that really fits the data is the one that accumulates evidences. So in this model, every time you get a new whiff, the probability to switch from the rate to switch from stop to go increases. And at some point you go. And you see that this model can basically replicate the data pretty well. And the duration of the stop depends on the whiff frequency again, and in the data also. So the message here is that once the fly is stopped, it doesn't go necessarily immediately at the first whiff. It accumulates evidence and then go. So this is basically what I wanted to tell you. We engineered this new assay, which allow us to precisely see the behavior of the fly and also the odor position by looking at the smoke. And from that, we found that in this complex environment, where the whiff duration is very short in our experiments, we're talking about 100 milliseconds, 50 milliseconds, and less. No, we see that the orientation upwind increases with the whiff frequency, but doesn't seem to depend a lot on the whiff durations. An interesting finding is that the fly seems to be instantiating terms at random time, but it biases those terms upwind as a function in response to high frequency of packets of odors. And the fly also do a lot of transition between stop and go. And they decide to stop if they haven't experienced a whiff for a little while. And the duration of time until they stop is exponentially distributed and only depend on the time of the last whiff they experience. But the decision to go is different. The decision to go depend on the sequence of whiff that they have experienced. So they accumulate information before they go. And with that, I'll finish, and I'm happy to take questions.
Thank you very much. This is a very interesting talk, and I think we have a number of questions specifically for your talk.
So let me move the camera here. Yeah.
Okay. So first question is from Rory. Great talk. In your smoke navigation essay, how well does what we see visually correlate with what the fly smells, i.e. react to? Is there evidence of flies reacting to invisible odor signals not represented by the visible plume?
Yeah, that's a very good question. Let me show how we... So obviously, when you have an experiment like that, you have to decide... can you see my screen here?
Yeah, outline. We see an outline slide.
Oh, was this... Somehow I went out of the So I have to do something. 1 second. How do I get out of this? Up, and I have to share again. Oh, that's annoying. Sorry about that.
No
I see
worries.
now I'm becoming an expert. All right, so you should see now something that's related to that question. You see it? Yes?
It could be, yeah.
Okay, so one thing that you have to do, so the first thing is that the smoke, it's a complex signal, and it has long carbon chains and so on, and what you see basically is just a part of it, no? And so we worried very much about how are we going to decide what is the detection level for a fly? What level of what we see do we say that the fly has experience with? No? And so to do that, what we did is that we looked at, so we took a movie here of a straight plume, and then so this is a zoom in of this, and so we have the level sets of the odor in gray here, and then we measure many flies navigating, and you see here those red dots are the moments when the flies are turning back towards the plume. Okay, and so we're assuming that at that moment the fly has kind of realized, okay, I lost the plume, or I lost the signal, no? And the plot below is basically reporting all of the turning points in smoke, and in black, all the turning point in ethyl acetate. So two different type of experiment. And then the red line, which is here, is a level set of the density of those points. And the pink lines is the smoke detection limit that we have in our experiment. And the blue line is the smoke threshold we chose for this experiment. And so the distance between here and there is on the order of about 1 millimeter. And in our assay, the wind is blowing at a very fast speed. And so we made the calculation that the error we're making in the timing of an odor arrival, or it's on the order of 10 milliseconds and so on. So this is how we basically decided when do we consider that the fly has entered a plume, and when does the fly has lost a plume. Okay.
Thank you. Let me see. Okay, a question from Matt. Have you varied the wind speed in your assay? Do you think the behavioral algorithm may adjust for that?
No, we haven't varied the wind speed here, because we kind of constrain by if you make the wind speed very slow, then you start to be in a different type of Peclet number. So you have to, we really wanted to study what happened when the fly is experiencing discrete packet of odor that pass fast. No, if you go to lower speed, then diffusion now becomes fast relative to the flow speed, and then you start to do very smooth plume. No? So you have a trade-off between the two. So we haven't done that.
I guess in some way you would expect the algorithm to be maybe quite different.
Yes, the behavior is going to be very different. And by the way, the behavior is quite different when we look at the straight plume. No? So in a straight plume, the fly does really almost deterministic zigzagging around the plume. It looks more like an ant than a. So we're very interested. Right now, we're studying basically how does the fly adapt its strategy to those different statistics of signal and environment. No?
Yeah, that's very interesting. I have another question from Dinesh. It is interesting to see that flies stop once the smoke stops. With odor, generally they cast for a while and then go downwind. it seems like this aspect is different between odor and smoke tracking. Any insights on why the difference?
So we get the same behavior exactly if we do the experiment with ethyl acetate. So it has nothing to do with the smoke. So we did that with ethyl acetate, with apple cider vinegar, and so on. So the flies really tend to stop a lot between when they lose the odor, and then they go downwind over longer time scales.
Okay. You see, I have there's some answers, some questions from earlier that Venki answered, so I'm skipping them for now. We can, if people want them to be asked again later, maybe just indicate. So you have another question from Ashley. Do you think this auto tracking work has implications on addiction work related to vapor exposure models, in particular on characterization of subsequent spatial behavior?
So I'm not familiar with the study. So vape. So what are those?
I can read it. Should I read it again also?
Maybe your.
So do you think the auto tracking work has implications on addiction work related to vapor exposure models? I also don't know. In particular on the characterization of subsequent spatial behavior. Maybe this is a good question for the broader.
Yeah, I don't know what vapor models are, but maybe it's related to whether if you change, for example, the Pecklet number, which is basically the ratio between the diffusivity and the inertia forces, no? So you can get smoother or more, let's say, intermittent plumes. So you can get a smoothing of the packets, or you can get packets that are sharper, depending on if the Pecklet number is low or high, no? And also, the presence of surfaces will cause the velocity to go down to zero at the surface, no? So there's a boundary layer effect that also will smooth out the odor profiles. So I think that the flies will experience many different types of statistics, and I think that this kind of experiment can study this broad range of statistics and how the flies can instantiate strategies in different environments. I don't know if that answers the question.
Yeah, we can later maybe ask Ashley to elaborate, and then we can. I think I have one more straightforward question that's very specific from Istvan. Do you know if the bias turning direction depends on intact ariste, that is wind sensing, or can flies make the correct turn just based on odor lateralization?
I don't know the answer to that, but this is on the list to do. So definitely, yes. That's a very interesting question.
Okay, maybe one last question. This is from Aaron. I'm curious how well pixel intensity correlates to odor concentration experienced by the flies, since the camera integrates over 3D Plume to some extent onto the image plane where the flies are imaged. Thanks and great talk.
Yeah, that's also a good question. I think that because of the geometry of the problem, so the flow is between two glasses that are separated by 1 centimeter. So really, I expect that in the vertical direction, you have a typical flow field that has this 0 at the boundaries and then maximal in the middle. And but yeah, we don't have a direct measurement of the profile. So we're really just integrating through this 1 centimeter. The other thing is that we also don't have a direct, scale. We don't have we don't know the odor concentration of the smoke. So what we only have is that we can relate monotonically the odor concentration to the opacity in the image that we're seeing. And we verified with the dose response that as you increase the odor and the opacity, you get more and more flies that accumulate around the center. But that's all what we have. We don't have a direct relationship between the concentration of the odor and the opacity. And that's one of the reasons also is because this odor is very complex. It's going to have CO2 and many, many different components in it. So we really just use it as an attractive odor that we can visualize so that we can zoom in on the time-dependent reaction of the behavior to the signal.
Okay, thank you. I mean, you can keep asking clarifying questions, but I'm going to bring in all the discussion panelists, and we can also broaden up the questions a little bit. Thierry, can you close the?
Oh yeah.
Or maybe I can. Okay, I can do it. I guess. Okay.
You kick me out of the box.
Yeah, I can do it. I guess. Right now, I'm trying to get everyone in. Sorry, it's gonna take me a moment. We got a lot of questions this time. Okay. Okay, I think. Maybe, so Dennis had unstable internet connection. I don't know. Okay. We'll see. There he is. So I think we could start. I try to remember exactly, but we had this sort of slightly branching into a broader topic question earlier, which was kind of, can we put these lab behaviors into ecological context? And I think it maybe relates a bit also to basically these the paradigms we saw, they take one slice of possible kind of stimulus, right? Like the perfectly, like the very fluctuating or the boundary condition case, etc. So maybe you have some thoughts on how this all comes together when you have an animal walking in or navigating in the wild.
Is that a question for both of us, I guess?
Yeah, it's for both of you. Yeah, sorry. So either one of you, go ahead.
I can give you my thoughts that are mostly useless, which is that I think until we have a sense of what is being sensed, it seems like just watching animals, I mean, this is a bit of a fantasy, right? That we can watch an animal behave and without really knowing what they're taking in, we can somehow make sense of it. Maybe that's true, but I'm not so sure that we'll ever get enough data for that to be true. So it seems like you really do need, and that becomes much harder in the olfactory system, and things like, I think Matt Smear said, like, what is the natural statistics? You never even mind the specific instantiation with Thierry. Thierry's experiments, one of the beauties is that you really know from millisecond to millisecond the distribution of the odor at some resolution. So I think I'm very sympathetic to the, like, we want to know what's happening in the wild, but if it's just a matter of just watching what's happening in the wild, I personally am not that interested. Because that's been done and we can do more of that, but it seems like without knowing what's being sensed and what that, I just feel like it's unclear. So anyway, that's neither here nor there, but maybe Thierry has other thoughts.
Yeah, I think I think there are two sides to me. One is that in the world, in the wild, you can have different time scale, length scales, and intensity scales. statistics, and you see this type of statistics in many different places, and you see these different scales or in many places. You can look at fishes or crabs, you can look at dogs, you can look at mice, you can look at ants, you can look at flies, and in every cases there's going to be a range of the statistics of the environment over which that animal is absolutely able to navigate. But that range is typically very wide, and I suspect that an animal will use different strategy in the different ranges. And so by the time you add all of the data from all of the animals together, you actually get like a very wide range of statistics and behavior. And one thing that's interesting is that when you start to look at the overlap between, so what two different animals do in the same range, if you want, it's not that different. There's a lot of things that get conserved. And I know that because I work with bacteria, which are, you know, not as intelligent as flies, although maybe we could argue about that just for the sake of it. I mean, the fly has a brain, no? But those E. coli are amazing. They can navigate complex environment also, and with much less hardware than the fly, no? And they are instantiation of the behavior that I see in the fly and that I see in the bacteria that are similar. And I think it boils down to length scales, time scales over which you access information and you're able to use the information. And so in that sense, I think that there is something to be said about studying all of those different behavior and really quantify from a physics point of view those constraints. And then it becomes easier to understand why an animal would do something than another one if you understand the constraint. And then the model systems are great because you can, for example, we have developed an optogenetic assay where we can do the same plume but with light. And so we're using that now, and we can, in that context, you can do all sorts of things that you could not do with a real odor. So model system like a fly, it's fantastic to be able to then dissect, okay, what information is the fly brain receiving? Can I manipulate that, etc.? And I think with the mice, like the experiment that Venki is building, the same thing. Amazing, you know, with the treadmill, you can do, there's so many questions you can ask. So I think that my take on the answer to the question is very positive. Like we're in the right moment now to break this problem.
I just want to correct myself to say, like, I didn't mean that you shouldn't. Obviously, you should study behavior in the wild. I mean, this is, you know, I should be quartered, right, if I disagreed with that. I think it's more that I just don't know how you can ascribe specific moves and turns and whatnot to an olfactory stimulus. In the wild, you have many sensory modalities. That's what I meant. That I think in our case, we are taking care to change the odor olfactory stimuli so you can do this kind of relation. But out in the wild, I don't know if the mouse did a sudden jump. Is it because of what? So I think that's what I meant, just to be very precise. And Matt Smear jokingly put in a field deep lab cut. Yes, I think we should be doing post detection and behavioral automated behavior, which I think many people are already. So I am with Thierry that I'm optimistic, but I just meant your answer question of, like, should we just ascribe olfactory behaviors out in the wild by just watching them? I don't know about that.
Yeah, no, I actually agree with that. But one quick follow-up question then is, do you think the nice thing about these pretty defined paradigms that you presented is, I mean, you can break down the behavior to quite a high level of detail. So do you think if you then look at basically in a complex setting and maybe in the wild even, could you say, could you identify from even the behavior that probably they're using strategy X that makes sense because of these statistics in the environment? Or do you think that's also not feasible because it's just too, I don't know, high dimension?
It's a great point. looking for patterns, I think you're saying, and then you study them. If that's, then you can go on. I was also going to make another point, which is that, you know, there aren't very many experiments where you can, if there are ways to look in the wild to really have a strong basis to say, yes, they're using the sense of smell to do such and such, right? I mean, you know, this, for example, you know, there is this beautiful studies the field, like, you know, albatrosses, and, you know, we all know, of course, the story about salmon going up. So it's all there. I think there are very suggested many different things, but what do us want are the moths, right, in the insect field? That's, you know, it's an absolutely beautiful history. So I'm just very jealous in mice. Of course, you know, they have an amazing olfactory system, but if somebody asks me, like, what is the natural, give me an example and explain that in detail, what is that behavior, it's a little embarrassing.
Unless Thierry wanted, I wanted to, sorry, I failed to probably introduce Chris earlier, and maybe you want to briefly present yourself, and if you have questions, you should go ahead and ask.
Thanks. Yeah, I'm Chris. I wanted to give a brief shout out to the whiskers field, which is where I work, because I thought there were some really cool links, conceptual links and parallels to what we saw today. You know, in particular, the whiskers have to be actively moved to collect information, similar to the kinds of motor actions that we saw. And that when they do, so, you know, following up on Venki's point about the difficulty of knowing exactly what the animal is sensing, I think that's one of the, in other modalities, this can be a little bit easier. We can see exactly what the whiskers are touching and have a sense of the input to the system. And when we've done that, we actually see something that I think is really similar to what each of you presented today. We see that the sensory information arrives in these really sparse packets, and that the relative prevalence of those packets could be really important for driving the animal's decision. We have a paper coming out soon about that. So I think it's really cool to see some kind of common computational algorithms there. I guess I wanted to ask a question. I was really liked one of Venki's early slides where he talked about in the moment or reactive strategies versus more planning based strategies. And I was wondering at what level we would need to see. I think one thing we've seen is that an in the moment strategy can get you a really long way in a lot of situations. And it's actually kind of hard to point to something where you really need a more planning based strategy. And I wonder, at what point does an internal model become necessary? That the mouse, you know, for instance, in your long distance odor behavior, which you only presented the head fixed kind of stripped down version of that, but I imagine it might have to build up an internal model of the odor, the whiff frequency at all these different locations in space in order to determine that gradient. Do you think that something like that might require an internal model? And if so, sort of what brain structures do you think might be involved in that?
Excellent question. So as you well know, I mean, this is kind of the big thing, and people talk about this quite a lot in the motor systems, have been for a long time. And it's, you know, it has various names, like, you know, if you're doing deep nets or something like that, you call it generative models. And so this idea of building the model in your head is very cool. And I think for mice, I don't know, I would take a large bet that yes, they're going to have something like that. I don't know how complex it is. Also wanted to point out, and I think I'm sure Thierry understands this mathematically much more than I do, but the infotaxis algorithm in some sense does have that. It has essentially all the conditional probabilities moving back in a long time. So that's effectively an internal model of the, you know, you need to know how the world is. So I think for our ant experiments, I think just to give you specific examples, one experiment that we've been doing, but we've been doing always slightly wrong for you know, over many months now, that if you give us a series of trails, a sequence of trail, right, a long trail with specific statistics, and then break it, right? So how much is it built up? Like, does it know that it's curving sinusoidally at a certain curvature for some, right? So that can give you, and you can call it a time constant, but it's also in some way an internal model. It's not a map, but it's a, for mice, I think, you know, once we have experience in that for mice, we would love to do something like that, right? It's maybe more than just your adaptation to the statistic for the last 5 seconds, but maybe there's something more over maybe even days. will they kind of know that if I get this and I'm going to have a fork, or if I get this. So I think those are things at the moment in my head, conceptual, that these experiments. But I would take a large bet in some well-defined way there will be an internal model of this trail tracking space with that. But I don't know what the theory thinks, because the theory taught a lot about adaptation, right? That's in a way that's also history in some way.
Actually, yeah. So, I think the value of very, very simple model is especially important in asking these questions. Like, for example, following a trail or doing something else, because you can start to ask question of how does the behavior depend on the statistics of the type of signal. And if you go even simpler in actual bacterial chemotaxis again, we discovered something that I think is, I would call it the smallest possible model you could make of the world, in the sense that the strategy of the bacterium is super simple. No, it's I'm instantiating random turn, and so I have a probability to turn, and whenever life is getting better, I lower that probability to turn. That's the model. But if you think about it, whenever you navigate in the right direction, you're going to lower the probability to turn. So that will cause you to, on average, move further up in the right direction, which will further lower your internal state of probability to change direction. And what we discover is that there's a positive feedback of your decision on the signal, and that is key. And so in any navigation problem, whenever you make the right decisions, you get more information about the location. You get more informative information from your sensors if you do the right moves, in some sense. And that has a positive feedback effect that drives your internal state away from a steady state of quasi-equilibrium, if you want. So in the bacterium, it's a very, very simple thing. You have a probability to change direction, but when you start to climb gradient, what effectively you see after a few seconds, that probability is lowered. And so the cell is not anymore a cell that tumbles 20% of the time. It's now a cell that tumbles 10% of the time. And it climbs much, much faster the gradient. Actually, so fast that you see cells climbing like a ratchet. And you can show that, the benefit in performance are enormous just because of that. So I think that, going after that question is, first of all, the most interesting, one of the most interesting questions to me. And second, that you can start with very, very simple. If you start the simplest possible is the best way, because if not, it's going to be super complicated. And so, for example, Venki's approach with the trail, doing that would be great. And we're trying to do something like that with projecting light in those arena, and basically playing with those statistics also, and see if there's a history dependence on the navigation.
Can I quickly ask Chris a question about since you mentioned whiskers, it's on the table. So one of the things that we get asked, and we ourselves haven't done studies and some other stuff. So in mice, and I think since NF Maximir is still on that clearly they're sensing the wind too in these conditions, or at least for the airborne kind of stuff. Do you have a sense that is it pretty clear that there's something like wind-guided or odor-guided animotaxis
in the whisker system?
Well, yeah, so the whiskers, just like flies, that seems to be insects. It seems to be a pretty simple thing, right? That you smell odors, but you're using a lot of the wind sensing to. And I don't. It's only one or two papers in mice where it's pretty clear that they're sensing wind, but I don't know what you sense that is that going to be used for odor-guided navigation.
Well, so Mitra Hartman's lab has shown, maybe that's what you're referring to. So she's definitely shown, that's the only one I know about too, that's shown that the whisker system is important for detecting the direction of airflow. Yeah. And I think that I mean, I think in general, a lot of these things are going to be multimodal. I think that you were pointing out, like, oh, do we really have any clear examples of olfaction in the wild driving something? Well, I have a ton of examples in the lab of tasks that were supposed to be whisker guided and it ended up being olfactory guided. And we see these things as kind of, in the lab, we want everything to be a single modality. I want to just be studying, the C1 whisker. You want to study something that's purely olfactory. And I think that's going to be the challenge in the wild, that it's going to be hard to say this is a purely olfactory behavior, because it's probably almost always going to involve multiple modalities, such as wind, such as olfactory cues. And I think, we talked a little bit, it came up after your talk, we talked about detecting, is trail following even something that we know that mice can do? I thought that was mainly the case actually for pheromones in the auxiliary olfactory system, the non-air, the chemicals that are detected by surface contact rather than by whiffs. And I thought that in that case, it was pretty clear that they do this kind of like surface detection.
I think it's that's kind of becoming more and more confusing. My next door neighbor is Catherine Duloc, who studied this quite a long time. So it's very clear that what many cues that are thought to be pheromonal are going through the main olfactory system and vice versa. And the pheromone system, I think, needs contact just for like the way it accesses the physical stimuli. Like it has this little pump thing that has to send the stuff. So you almost cannot do it with the airborne kind of cue. But there too, I don't know if it's necessarily trail tracking versus whatnot. But this is a great question. We all immediately think of dogs, right, when we talk about trail tracking, and nobody doubts that. At least certain kinds of dogs. I would be curious to think of experiments out in the wild or just watching in the wild to see if.
I think there's also a time scale again. So dogs do two things. No, they lift their head, they smell, or they sniff again and again out on the floor, and they definitely interpret that very differently. Like they lift the head to get the sense of what's far away and being transported. So it's this one type of information, and they look at the floor and they sniff the floor to detect who was there maybe 2 days ago. So there's a history dependency of the trail that airborne cues don't have. I mean, airborne cues now, the floor is, history. And actually, so the first paper I ever did in olfaction and in neuroscience ever was to study the response of olfactory receptor neuron. And the first thing I did was use a PID, which in those days people didn't use, to measure the odor signal. And then I realized that the odor signal had vastly different temporal dynamics in the standard lab experiment. And that most of the neuronal response were basically a consequence of a convolution between the signal that was different for different odors, even though people were assuming that it was the same. And so in odor sticks to surfaces, and they stick to surfaces depending on their physical chemical properties. And that is an entire world of differences. So I'm sure that animals know how to interpret the differences in the temporal information. It's like when you go into a room and you smell cigarette, it could be cigarette from a day before. But if you smell, let's say, some lemon, it must be.
That reminds me too of another idea about whiskers and olfaction being related. This is total speculation, I think, but David Kleinfeld has studied these links between the motion of the head, as they raise their head and lower their head, similar to what you were just saying, Thierry, and also the sniff cycle and the whiskers. These are all locked oscillators that move together. And that part is very rigorously shown. What he was speculating about is that they might be using the whiskers to sweep along the floor to like pull up all of the olfactory molecules that are trapped on the floor. They could sweep them up with their whiskers and then lift their head and smell it. And I think that's something that I could totally believe happening in the wild, but I think will be extremely difficult to actually I don't know, get a PID down there or measure like what they're sweeping up with their whiskers. But it's a cool idea.
Sorry, like I feel really bad, but I think we need to wrap up soon. We're kind of over time, but I mean, we got so much engagement, so I think it shows this was a really good session, and like thank you very much for really great talks. And
thanks for the invitation.
Thank you for inviting us.
Yeah.
I hope we could. I wish we could hang out.
Yeah, this is very awkward. When I close this, we'll like just disappear. We're thinking about some kind of opportunity in the future to maybe have a more informal discussion, but we have to figure out logistics and stuff. But yeah, so
now we're all vaccinated, so we should be able to talk for real.
He wants to go to Lisbon so desperately.
I understand. We all want to go to Lisbon. All right.
See you all.
Thank you very much. Bye-bye.
Yeah, see you soon, hopefully. Bye-bye.